So bile secretions is the topic for this short video. The secretion of bile is the main digestive and absorptive contribution of the liver to the GI tract. It's very important for regulating metabolism, many other processes in the body, but as far as digestion and absorption, the secretion of bile products is how the liver contributes to this process. The components of bile are several. You have bile salts, which are conjugates of bile acids. You have bile pigments, bilirubin. Cholesterol is a precursor of bile acids, and so that's found there. You have phospholipids, proteins, and electrolytes. And the composition of bile is isotonic with the plasma, and you make about half a litre to 1,200 mils a day. And the bile acids are synthesized by hepatocytes from cholesterol, like I mentioned. You have primary bile acids like cholic acid and echinodeoxycholic acid, and these are formed in the liver and secreted, and then as the bile acids pass through the lumen of the GI tract, they're affected by bacteria and dehydroxylated, and this creates secondary bile acids like deoxycholic acid and lithocholic acid. And bile contains both primary and secondary bile acids. This is how the cholesterol is changed into cholic acid, and echinodeoxycholic acid is hydroxylated in this position, and these are the primary bile acids, and then intestinal bacteria will then dehydroxylate these things and create the secondary bile acids. So the function of bile is to create bile salts that is important for absorption of lipids in the small intestines. These will emulsify the lipids and form micelles that are necessary for lipid digestion and absorption. So these micelles will contain lipids and bile salts and enzymes that then degrade the lipids down into monoglycerates and fatty acids ready for absorption. And these conjugated bile salts are the product of the combination of the bile acids with either glycine or taurine, and it creates this peptide linkage conjugated, and these then are the bile salts. The bile acids are derived from cholesterol, and therefore this is the only pathway that cholesterol can be secreted. And we'll see that there's this whole sort of bile salt flow where it's secreted and then reabsorbed, but some of it invariably then gets excreted out with the feces, and this is the only pathway that your body has to get rid of cholesterol. And also the function of bile is the excretion of bilirubin, which is a major product of hemoglobin degradation, and passing this into the GI tract is one way that the body excretes this substance. So this is the circulation that I briefly mentioned, and about 95% of bile acids are recirculated. So they're created in the liver and then stored in the gallbladder and then secreted into the duodenum for the emulsification and digestion of fats through micelle formation and fat absorption. And then as it gets down into the terminal ileum, or even in the proximal colon, these bile acids are reabsorbed, and through the portal circulation they're then taken back into the liver, where they're acted on, either reconjugated or hydroxylated to form primary bile acids and salts. So the main pathway for the absorption of bile acids is through active reabsorption of the conjugated bile salts. And like I said, this happens in the terminal ileum. and it's a sodium coupled transporter. The secondary pathway then is passive absorption of unconjugated bile salts, and the difference between the unconjugated and the conjugated is their solubility. Conjugated bile salts tend to be more soluble than unconjugated, and this is why they need a transporter to pass across, but the unconjugated bile acids are very lipophilic and will just passively diffuse across the apical membrane of the epithelial cells. Bacteria in the terminal ileum and colon deconjugate the bile salts, and this then promotes the passive absorption of these deconjugated moieties. Bacteria also dehydroxylate primary bile acids to create secondary bile acids, as mentioned previously. And so this is the facilitated transport of conjugated bile salts into the hepatic portal circulation. And so you have sodium comes in together with a conjugated bile acid, and then these will be bound to cellular proteins and transported into the portal blood supply. As you see, the unconjugated bile acids just passively diffuse across the membrane, being so lipophilic, and then they enter into the same kind of pathways and transportation back to the liver. So once the bile acids and conjugates return to the liver via the portal blood supply, they're reuptaken by hepatocytes and then primed for resecretion. Unconjugated bile acids are reconjugated, and secondary bile acids are then rehydroxylated into primary acids, and they're primed then ready for resecretion. So one of the main products in bile, and it's responsible for its color, this kind of green bile color, is bilirubin, and this is a product of hemoglobin degradation. And so when hemoglobin is broken up, you get globin, which is a large protein molecule that's digested and enters into the free amino acid pool of the body. But the heme part then is separated, the iron is reutilized for other hemoglobin creation and just goes into the iron pool of the body. But the other part of it then is bilirubin, and then this is passed out into the GI tract via bile secretions. This is a more detail of that metabolism of bilirubin. So once you get the breakdown of hemoglobin, you have biliverdin that becomes bilirubin, and then this is transported through the blood bound to albumin, and then it's taken up by the hepatocytes and conjugated, and then passed out into the small intestine, and then it's created. It forms either urobilinogen and secreted out through the urine, and this gives urine its characteristic yellow color, or it forms stercobilin, and then this is excreted out in the feces, and the color of the feces is predominantly due to the presence of stercobilin. If you have too much bilirubin production, then you get jaundice, which is the yellowing of the skin, and you can see this in sort of the whites of the eyes and just the general tone of the skin. And this is seen when the total plasma concentration of bilirubin is above 2 mg per deciliter, and this is hyperbilirubinemia. And the causes of these are numerous. You could have excess production of bilirubin because of excessive breakdown of red blood cells, and this is called hemolytic anemia. Or there could be problems with the liver, whether you get decreased uptake of bilirubin into hepatic cells, and so you get this large circulating concentrations that you can see from outside the body. There could be disturbed intracellular protein binding and conjugation, or disturbed secretion of conjugated bilirubin into the bile caliculi, and there are several diseases that cause that. Or there could be a blockage in the hepatic and extrahepatic bile ducts, causing a sort of feedback then that builds up the bilirubin in the blood, and the symptoms and signs of jaundice are then seen. So the control of biliary secretions, it's dependent on how much bile salts are in the hepatic portal blood. And if the liver detects that there's not enough, then it's going to start synthesizing bile salts. It's also under hormonal control. Cholecystokinin is the main hormone that promotes secretion and creation of bile salts, and gastrin has a minor role. And then it's obviously under neuronal control, similar to the scheme of the general gastrointestinal tract. Parasympathetic stimulates the secretion of biliary juices, and then the sympathetic stimulation will decrease that.